Hello everyone and welcome to this class. In this class, we are going to study about food preservation methods, and in those food preservation methods, we are basically going to learn about physical methods of food preservation. Okay, so let us continue. So first of all, we will start with the question that what do you mean by food preservation? Okay, so before starting this, I would like to clear that the food preservation methods were prevailing in the world from ancient times actually. And with that, we are going to start the food preservation. Okay. So what is this food preservation? So food preservation is the process of treating and handling the food to prevent spoilage or and to prolong its shelf life. And this also by maintaining its nutritional value, texture, and flavor too. Okay. So the goal of food preservation is to slow down or stop the process of microbial growth, such as bacteria, or you can say yeast or molds, whichever that you know oxidizes or spoils the food. Because the food preservation prevents the oxidation, and it also minimizes the chemical changes in the food, which leads to preservation and stop the deterioration of the food. Okay. So here are the various techniques which can be used for food preservation. The first will be your refrigeration. Second is your canning. Third is your drying. And fourth will be your fermentation. The use of preservatives are employed to ensure that food remains safe and edible for extended period of time. Okay. What is the basic principle behind the preservation of food? The basic principle behind the preservation of the food is basically to control the factors and microorganisms that can lead to the spoilage of food. By stopping them, we can stop the spoilage, and thus the food is, uh, you know, remain in its normal condition or natural condition, in which it is edible and safe to consume. So the factors which are required or which we can control are temperature, moisture, pH, oxygen availability, and chemical additives. Okay. First of all, we will start with the temperature. Now, as this is a part of microbiology, so I'm going to talk here with respect to microorganisms. Okay. So, in case of temperature, like microorganisms need good temperature. Okay. So, just like our body, 37 degrees Celsius is the optimum temperature for microorganisms to grow. So, if you keep your food around this temperature, then it is going to spoil fast. So, what we can do is we can decrease the temperature. Okay, decrease the temperature, and after decreasing the temperature, what happens is the growth of the microorganism also decreases because their metabolic rate, it slows down, or they almost go to the dormant state. Okay. Then we have the point of moisture, which is our second point. Okay. So in case of moisture, what happens is the moisture, which means basically water in its gaseous form, is required by microorganisms to grow. So there is an optimum moisture content which is required by microorganisms. So what we can do is we can control the humidity or moisture in the surrounding, that can also increase the shelf life or decrease the spoilage of food. Then we have pH, right? So we take the example of neutrophiles. Neutrophiles. So we can say neutrophiles are the class of bacteria which grow around the pH 7. Then we have acidophiles which grow around the pH of 1, matlab toward 1. And then we have alkalophiles, which grow above 7 or in 8, 9, 10 pH, like that. So by basically uh, adjusting the pH, we can also increase the shelf life of our food. Okay. So if you talk here, like if normal bacteria, normal bacteria which grow at pH 7, if we decrease the pH, okay, like we are pickling something, like we are pickling the cucumber, we are pickling some veggies, that decreases the overall pH of the solution or of the food actually. and by doing that we get a little acidic flavor, which is sour flavor, and also we preserve that thing, or what we can say is that food, from being spoiled. Then we have oxygen availability. Okay, now just like water content, oxygen is also needed by basically anaerobic bacteria. Sorry, not anaerobic but aerobic bacteria. So aerobic bacteria will utilize oxygen. So if we cut the oxygen supply to our food by, we can just can it, right? Or we can seal the food. That can also prevent the spoilage of food. Then we have some chemical additives which we can also add to the food. These are what you usually call preservatives. Okay. Now in this class we are basically going to learn about the preservation methods of food via physical methods. Okay. So first we have dehydration. Dehydration is basically drying, or you can say H2O removal. Okay. So in this case, what happens is, it is the process of removing water from food, which reduces the water activity and thus inhibit the growth of microorganisms. Now, what is the principle behind this drying is, by reducing the moisture content from the environment or from the food, we create an unfavorable condition for microorganisms. So microorganism will not grow. Okay. So this is what happens. Okay. Now we will move to the types of drying which we usually use. So first will be your sun drying. So sun drying is basically drying any food or any uh, eatable substance in sun, right? Like if you take the example of green chili, we basically dry them. We dry them under the sun, and they, although they become red, but they retain their spiciness and their quality which we consume. Similarly, you know the case of papad. A papad in India are very often eaten, and they are also a good example of sun drying. You sun dry them, then you pack them, and then they can be also available for sale. Then the third thing is air drying. Okay, in this air drying, we use hot air to evaporate water. Okay, so in drying, as you know, that most of the things we do is to remove the water. So air drying is similar to that. The third drying process is your spray drying. In spray drying, we use basically this is used for drying of the liquid food such as milk and juice. Okay, so the food is sprayed with hot air in a chamber. Okay, and then we dry out the water from it. Then the last process is what we call as drum drying, which is fourth here. Okay, one, two, and this is your fourth process, which is drum drying. In this, food is placed in hot rotating drum. Okay, basically drum, it is like a plate. Okay, and the food is placed over it. This drum rotates and rotates and also heated at the same time. Heat is given to this drum, and the food will stick uh, to this drum in the form of layer which can be later scrapped. This is your drum drying. Then we have freeze drying, or what you call as lyophilization. So here is the laboratory sized lyophilizer. This is your lyophilizer. So in this lyophilizer, this is the main chamber where we can add our samples. Okay, so you can either remove this. First you have to remove this upper cover. Okay, and then you can place your sample in your beaker or in any container inside this. Okay, not inside this hole, but basically you can place them on this steel area. Also we have these valves. Here you can attach your flasks which are specially made for this lyophilizer, and by doing that you can freeze dry your sample. Okay, so So the principle behind the freeze drying is that if you talk about the state of water, then uh, you know that water exists in three states: solid, liquid, and gaseous state. But there is a point in temperature where water can exist in all these three states. Okay. Now, if water exists in all these three states, then at that particular, if we decrease the temperature below that, then we basically freeze the water to to the solid state, right? But in that state, if we apply a vacuum, if we apply a vacuum, and we decrease, obviously we are decreasing the temperature. So this solid can directly turn into gas without turning into liquid, which is your process of sublimation. So this is the principle which is followed in lyophilization, and this is how you basically dry or can dry your food stuff. Okay. So here I have written that in this, I mean in lyophilization, you remove the water from any food by sublimation. Okay, sublimation is the directly converting solid into vapors or gaseous state. This gives the liquid state. So we remove water without heating. Therefore, flavor, structure, and nutrition content are preserved by using the method of lyophilization. Now, the third method which we can use to preserve our food is via using heat. Now, heat is used to incinerate, kill or incinerate the microorganisms. Basically, these incineration is basically, you know, flaming the food like that. Okay. So by doing that, we directly kill or inactivate the microorganism, or we can inactivate the enzymes that can cause the biological changes. The principle behind this is when we apply the heat, it denatures the enzymes, proteins of the microorganism, thereby destroying or inactivating them. Okay. So here are main three processes in heating which we use on kind of on daily basis. First will be your pasteurization. Pasteurization is commonly used in milk or dairy industry, where we give for 72 degrees Celsius temperature for 15 seconds, and that is enough to kill all the pathogenic microorganisms. Then the second is sterilization. Now sterilization, we are basically using autoclaving, in which we use the temperature of 121 degrees Celsius, and that is done for about 15 minutes. So this is basically our steam sterilization, which we use. Okay. Then the third is the blanching, which is quick heat treatment to inactivate the enzymes in fruits or vegetables before freezing and drying them. So blanching can be new to some. Basically, in this we use steam, or apart from steam, we use water, which is warm, or you can say hot water. Okay. First, we will dip the fruit or the food stuff, which can be done by this method, like we are fruits or vegetables. Okay, we dip them into the steam or water bath, and by doing that, many microorganisms still are being inactivated, and after that, we can freeze them or we can give them for drying. So further preservation can be done. The fourth will be your irradiation. Okay, so irradiation is exposing the food to ionizing radiation. Now, ionizing radiation can be X-rays, gamma rays, or can be electron beam. Now, what they do is they damage the DNA of microorganism, leading to the death of microorganism, and thereby no microorganisms means no food spoilage and food is preserved. That is it for this class. Thank you very much and